Fopius arisanus is an important biological control agent in Hawaii because it attacks tephritid fruit flies, which are serious economic pests throughout the tropics and especially here in Hawaii. The beauty of Fopius arisanus is that it attacks the egg stage of the pest insect, thereby controlling the insect at an early stage. In the following clips, you'll be able to see close-ups of how we rear Fopius arisanus in a laboratory setting. And at the end of this program, you'll see some discussions of how this particular parasitoid has been used in a successful release program in French Polynesia.
We will begin our protocol with adult parasites that are ready to oviposit into eggs of a host fruit fly. The first step is to prepare the fruit fly eggs for presentation to the parasite. Next, fruit fly larvae are allowed to hatch, develop, and pupate. Once the fruit flies reach the pupal stage, they are sorted by size to enrich for parasites. Finally, parasites emerge and are maintained until maturation. They then mate and are again ready to oviposit into a new batch of fruit fly eggs. This completes the cycle.
Prepare a substrate for the fruit fly eggs by making agar blocks approximately 10 centimeters per side. The agar is prepared at a concentration of 9 grams of agar powder per liter of water. Using a level surface, fill the dishes to the rim with liquid agar, allowing it to solidify for at least 45 minutes. Once the agar blocks are solid, apply